Hi everyone, my name is Piyush Kumar Pathak, and in this video we are going to discuss one of the very important topic that is in both biology and chemistry for NEET and some similar competitive examinations. So let's dive deep into the topic. So the name of the topic is biomolecules, and in this chapter I will discuss amino acids and proteins. So this is both in chemistry and both in biology, and so we will study it like we will cover both biology and chemistry. So let's start with the topic. Okay. So the first question is, what are amino acids? So what are amino acids? The name consists of two terms, that is amino and acid. So if you have a little knowledge of chemistry, you can easily understand that it would have an amine group and an acidic part, that is carboxyl group. So we go further into the chemistry of amino acids. First, let to take a brief of background of the amino acids. So amino acids are the building block of protein. They are chained together in to form proteins. They are linked to each other to form proteins. Okay. So basically there are many proteins. There are many proteins in nature. There are many proteins in nature, approximately 4 to 500, but they are present in nature. But in biological system, there are basically 20 amino acids are present. So 20 amino acids are present in biological. So 20 amino acids are present in biological system, and most of these amino acids are alpha L protein. We will talk about this, what is alpha and what is L amino acid. Sorry, but alpha L amino acid. What this alpha means, what this L means, we will understand these in further slides. Okay. So amino acids are building block of proteins. They are chained together to form proteins of various kinds. Basically, amino acids are classified into two categories, that are essential amino acids and non-essential amino acids. So these are essential. Anything that is essential to us means we cannot prepare it. Same is the case with amino acids. They are essential because our body cannot prepare them. We need to take them through our diet. So whatever protein supplements or proteins we take in our diet provides us with these essential amino acids. There can be also non-essential, but essential amino acids only comes through our diet. They cannot be prepared in our body. Okay. So basically there are 9 amino acids that are considered essential by scientists. These are methionine, threonine, histidine, valine, phenylalanine, tryptophan, isoleucine, leucine and lysine. So these are 9 essential amino acids that needs to be taken through the diet. They cannot be prepared in our body. Okay. We can learn it by a trick, that is PVT TIM HLL, private team hall. You can pronounce it at private team hall. So P stand for phenylalanine, P stand for phenylalanine, V stand for valine, V stand for valine, V stand for valine, T stand for tryptophan, this again T stands for threonine, I stand for isoleucine, M for methionine, H for histidine, L for leucine and L for lysine. So we can remember it was PVT Tim Hall. Okay, and let's talk about non-essential amino acids. So they are non-essential because they can be prepared by our own body through various metabolites that are intermediate of other reactions. They can be used to prepare these amino acids, and they need not to be taken through the diet. They can be prepared in our body, and the rest out of 29 are essential, so rest 11 will be non-essential. So 11 are non-essential amino acids. Okay, this is alanine, cysteine, aspartic acid, glutamic acid, glycine, asparagine, proline, glutamine, arginine, serine, and tyrosine. Okay, so this was the introduction of amino acid. Okay, now let's talk about the chemistry of amino acids. So in the chemistry we will first discuss the general structure of amino acid. Okay, so this is the general structure of amino acids. So basically amino acids consist of a central carbon through which there is one carboxyl group attached and one amine group is attached. One is hydrogen and this is R. This is R is side chain, or we can also say it as variable group. So this R group changes for different amino acids, and in this way there are different amino acids are formed. So in different amino acids this R group is different, and so there are different amino acids. Rest is same. So this is the generalized structure of a amino acids. So this is a generalized structure of amino acids. So you can understand this, that this group is different, this group is different, this is different, and if this one is different other than H, then all the attached groups to this carbon would be different, and thus it would be a chiral carbon. It would be a chiral carbon, and if this happens, then this amino acids can also show optical activity. This can show optical activity. Okay, this amino acid can show optical activity because if this R group is any other group other than hydrogen, then this all the groups would be different, and this carbon would be a chiral carbon, and it will be optically active. Okay, so now amino, there are, as I have told you, there are 20 amino acids. There are 20 amino acids. So these 20 amino acids are classified on various classification basis. There are various classification basis, but I will discuss some major classification basis on which these 20 amino acids are classified, on which these amino acids are classified before going into further details. Okay. So the first basis is non-polar side chain. As I have told you, this R group is different for different amino acids, and based on the nature of this R group, this classification basis is applied. Okay. So the first basis is non-polar side chain. The amino acids in which the side chain is, this side chain is non-polar. I mean there is no charge present on this side chain. Okay. So the first example is glycine. In case of glycine, the R group is replaced by, I mean the R group is hydrogen. The hydrogen is R group. And in this case, you can see that two groups are same. So only exception is glycine. Glycine is optically inactive. Optically inactive. Optically inactive. This glycine is optically inactive because here two same groups are present, and thus this carbon is not a chiral carbon. All other amino acids are chiral, have chiral carbon, but this glycine don't have a chiral carbon. Only this is exception. So we need to remember this point, that glycine is optically inactive. Rest are optically active. Okay, now let's move on to next example, that is alanine. In case of alanine, the side chain is a methyl group. Side chain is a methyl group, CH3. Okay, in case of valine, it's isopropyl. It's isopropyl. Okay, because the carbon is attached to the second carbon, so it's iso group, isopropyl. So isopropyl valine. In case of valine, the group is isopropyl. Similarly, in case of uh, Leucine, it's isobutyl because second carbon is attached to the chiral carbon. This all carbons are chiral. All carbons are chiral. So we don't need to remember all the examples and all the sites, and only some important ones are important. For example, in case of glycine, we need to remember that the side chain is hydrogen because this is an exception. It's optically inactive. So this one is important. Now this one is also important, methionine. In case of methionine, there is a sulfur group, and I will tell you the importance of sulfur group when I will discuss the protein part. Okay, sulfur group has free valency, so it can form bridges, sulfide bridges. So in case of antibodies also there are sulfide bridges. So sulfur groups, the amino acid that contains sulfur group are involved in formation of sulfide bridges. Sulfide. So this example is also important. Sulfide bridges. So sulfide bridges are formed by these sulfur. So all the amino acids that contain a sulfur group are important to remember. So one is methionine and another one is cysteine. Where is cysteine? Where is, yeah, this one. Look, here is also a sulfur. So methionine and cysteine are two amino acids that have sulfur group in them. So these are important. Next one is phenylalanine. Nothing important to remember about it. Just it has a aromatic. Is this benzene ring is present aromatic, so it shows aromaticity. Next one is tryptophan. This one is also important because tryptophan is the most heaviest. Tryptophan is most heaviest. It is the most heaviest, most complex, and because it has this large aromatic group, and this one is also aromatic. This one is also aromatic. So it will show aromaticity, and it is used in quantitative assay. This one is used in quantitative. This property helps in quantitative assay in protein estimation. In protein estimation, this tryptophan and proline also. This tryptophan and proline helps in quantitative assay of proteins. So in proteins that have tryptophan and proline, this would help us in quantitative assay. Okay, so both this tryptophan is important because it absorbs light and helps in quantitative assay. In a spectrophotometer, this one is important. Okay, anyways, let's move on to proline. So proline is also complex. Next we have serine. This one is also important. Serine has an OH group. And why this OH group is important? Because it helps in this one. Sorry, next classification is the groups that have polar side chain. So polar side chains, and polar side chains, that means they have polarity. Polar side chains means they have polarity. So that side chain will be, so this my here is, in case of OH, oxygen is more electronegative than hydrogen, so there would be a negative charge, partial negative, and there will be a partial positive charge. So this one is a polar group. This OH is polar group, so the amino acid will also be polar. Okay, so this serine is important because there is an OH group present. Similarly, one amino acid also has OH group, that is tyrosine. There is also a OH group. So these are OH group, alcohol containing, alcoholic group. OH is what? Alcohol. Okay, this is the functional group for alcohol. So these two amino acids contain OH group, and these are also important to remember. So I am writing the important ones. The first one is glycine. Second one to important is to remember is methionine and cysteine. The third one is tryptophan. Next one is tryptophan and phenylalanine. These are aromatic and helps in quantitative assay. Next one is serine. Serine is also important because it is at OH group. Another one is tyrosine. Tyrosine is also important because it has OH group. Okay. Next is asparagine, glutamine. Nothing important to remember about these one. And now the last basis of classification, that is electrically charged. Here polarity is not independent, charge is present. Polarity is different. Polarity means there is not a complete charge, but in case of this classification they have a complete charge because there are extra carboxyl and amine groups. So those amino acids that have an extra carboxylic acid, these are acidic amino acid, and those that have extra amine group, then they are basic amino acids. Okay. And so there are two acidic one, that is aspartic acid and glutamic acid. So aspartate and glutamate. But why there is not aspartic acid is aspartate? Because when hydrogen is lost from carboxyl group, it's become, now it was acid, now it's become it. The suffix changes. In chemistry you might have learned this. For example, so in chemistry you might have learned this. So if H is lost, then it becomes an conjugate base. So it is a conjugate base. Okay, so then suffix changes from acid to ate. Okay, so this is aspartate, this is glutamate. Okay, the next is basic amino acids that have an extra amine group. You see here there is an extra amine group, there is an extra O carboxyl group. Okay, here it has extra amine group, there is extra amine group. So these amino acids have an extra amine group and thus they are basic in nature, and these are acidic in nature. So these are also important to remember. Aspartic acid and glutamic acid. Okay, and next one is lysine. Lysine and lysine, arginine and histidine. So these are basic amino acids. Some important points are also that you will learn in cell chapter that when you will learn the nucleosome model, you will learn that the core is positively charged and DNA wraps on the nucleosome model. So the positive charge of the core is due to the presence of basic amino acids. Basic amino acids are present in large amount in the core of the nucleosome, that is the uh, what you say, histone octamer. And those basic amino acids are arginine and lysine. Arginine and lysine. So arginine and lysine are basic amino acids and these are present in the core of histone octamer and provides them positive charge. And since it has positive charge, the DNA has negative charge, so DNA can wrap on it due to electrostatic attraction. Okay, so this was a bit more information on amino acid. And next, move on to the next topic. Okay, so now as you see, the name of amino acids are quite difficult to remember, and they will also occupy a space. If you want to write a sequence of amino acids, it will take much space. So to simplify this problem, every amino acid has been assigned a single letter code and triple letter code. Okay, every amino acid has been assigned a one letter code and three letter code. Depend on the ease, we use any one of these. So I have arranged them in alphabetical order. Obviously some alphabets are missing because there are no amino acids for them. Okay, so we have A, not B, C, D, E, F, G, H, I, J, K, L, M, N, O is absent, P, Q, R, S, T, U is absent, V, W, X is absent, Y, and Z is absent. So basically we have 20 amino acids and 26 alphabets. So 6 alphabets are not present here. You not need to remember this, just need to understand this. If you have anything like ALA, so you can recall that this ALA is basically alanine. So if you have CYS, then you can remember that, oh, this is cysteine. Similarly, here ASP, aspartic. So these three-letter codes will be used in the NCRT book many a times. So you can recall by these codes that what are these amino acids, and this will be used somewhere in chemistry. This single-letter code. Okay. So this is the single-letter code and three-letter code for the amino acids. Now let's on to a very important topic, that is isoelectric pH. This question has been asked many times in NEET. What is isoelectric pH? Okay. So we have learned in the structure of amino acid that there is a amine group and there is a carboxyl group. Okay. So these are not present like this. These are basically present like this. Okay. These are basically present like this. There is a positive charge, the amine group receives an extra hydrogen, and this carboxyl group loses an hydrogen and it becomes negatively charged and it has a positive charge. So amino acids are basically present in this form, and this form of any molecule that has an internal positive and internal negative charge, then those types of molecules are called as Zwitter ions. Zwitter ion, you will learn in chemistry many Zwitter ions, but in biology this is also a Zwitter ion because this has an internal positive and internal negative charge. This form of any molecule is called its Zwitter ion form. And so what is isoelectric pH? As you see that there is a charged form of this amino acids. So if we apply electric field, if we apply any electric field, suppose we have a flask. Okay, suppose we have a flask. We have a flask and we have amino acid solution in it. And if we apply charge electric field to this solution, the amino acids will start moving towards any towards any electrode. Okay, towards any electrode. Obviously this solution will have a certain pH. This solution will have a certain pH. So the pH at which the internal charge of this Zwitter ion, remember the pH, we talk about pH in isoelectric point. Isoelectric point, so this is not any point, this is a pH. So isoelectric point is a pH at which there is no net movement of the amino acid in applied electric field. So if we apply electric field on the solution and the amino acids present in this solution do not move towards any electrode, then the pH of the solution is the isoelectric pH, isoelectric point of that particular amino acid. Isoelectric point of that particular amino acid. Okay, so you can understand that every amino acid has its own isoelectric point and these are their characteristics. So if I define isoelectric point, then pH at which pH at which amino acid has no net movement no net movement in applied electric applied electric field. Okay, so isoelectric point is a pH at which the amino acids has no net movement. And why this is happens, and why this happens? Because the charge is internally compensated. This is a very important point. Why there is no net movement? Because charge is internally compensated, internally compensated. So the charge is internally compensated, so there is no net movement towards any electric field, and this is called isoelectric point. So different amino acids have different isoelectric point, and this is their characteristics, and we use this characteristics to segregate amino acids. So we can, if we have a mixture of different amino acids, we can segregate them on the basis of their isoelectric point. Okay, so the next question arises is, what if the pH in which the amino acids are present is not isoelectric point? What will happen? So if the pH is not isoelectric point, then there will be a net movement. So this is the graph. So this is a basically a diagram. So this pH, this central pH is isoelectric pH. And if we have a pH lower than this, and if we have a pH higher than this. So if we have a pH lower than the isoelectric pH, then what will happen? So if we have a lower pH, then we have extra H plus in, so we have extra H plus in the medium, because pH is less than the isoelectric pH. So what will happen? The amine group will gain an extra hydrogen, okay, extra proton from the solution, and it will become positively charged. But since it has extra H plus, so here what will happen? There will be a similar effect like common ion effect. This one is weak base, so this one is weak acid, so it will not segregate in the presence of a strong acid that are already present in the solution. So here common ion effect, similar, something similar like that will happen, and this one won't segregate, but this one dissociate, but this one will get extra electron. So we see that in pH less than the isoelectric pH, we have positive charge on the amino acid. Similarly, if we have high pH than the isoelectric pH, what will happen? There is less hydrogen in the less proton in the solution. So what will happen? This carboxyl group will dissociate and release protons, and it will become positively charged, sorry, negatively charged. But amino group won't accept any hydrogen proton, because protons are less in the solution. So in pH higher than isoelectric point, the amino acid have a net, so amino acid have a net negative charge, and in pH lower than the isoelectric point, the amino acid have a net positive charge. So this can be asked in the exam. If they can ask isoelectric point, they can also ask this question. Okay, so we will keep this video up to here, and in the next video we will talk about the protein, their structure, the factor affecting protein structure, and many other things. Okay, thank you.